Welcome everyone. We're now going to start the second module of the Browsing Genes and Genomes with Ensembl course. So just before we start, I wanted to share the training page with you. I'll share this on the chat over here. And this is the page where you can find the course book, also the PDF version of the slides that I will be presenting, which I presented yesterday, today, and also during the course within this week. And also wanted to bring your attention to the living document. Some of you have been using the living document, so it has been quite busy, which is very nice to see. But the living document you can find on the training page over here. And if I just click on it, I'll just show you that we have a little bit of the introduction, and then we have space for questions. So I'm going to go back to my presentation now, and let me just switch off my camera and go into presentation mode. So yesterday we covered Introduction to Ensembl, and today I'm going to cover genes and transcripts in Ensembl. And then we also have the course overview for the next few days, which is we're going to cover Ensembl variation and comparative genomics, Ensembl regulation, and then next week on the 26th, I'm going to cover biomart. I mentioned yesterday that a gene in Ensembl is a collection of alternatively spliced transcripts. And on my slide, I'm showing you a region in detail view, and we explored this view in our demos yesterday, and also you must have seen it, you must have come across it whilst you were doing the exercises. And on this slide, I have different transcripts of a gene. So as I mentioned, that gene is a collection of transcripts, and it's absolutely normal for alternative transcripts to be transcribed from a single locus. They're also known as isoforms or splice variants, but personally, we're not a fan of splice variant term, as it implies that alternative splicing is the sole reason for alternative transcripts. However, it's more complicated than that. There are alternative transcription start sites and end sites, and there are alternative translation initiation sites. So just bear this in mind. All of the transcripts are biologically valid and real, and they could be expressed differently. They could serve different biological functions. So if we focus on a transcript on my slide over here, you can see that these are color-coded. So we can distinguish between protein coding genes. So these are genes which produce proteins and are translated. And then we also have non-coding genes which do not result in a protein. And these are like genes like RNA genes. We also have protein coding genes. Those can have protein coding transcripts, which are translated, and these are shown in red. But protein coding genes might also have non-coding transcripts, and these do not get translated. So those are, for example, retained intron transcripts or nonsense mediated decay. So these could be fragmentary. So they might be missing translation start sites, or they're not translated and they might be faulty, or they get degraded before they get translated. And these non-coding transcripts are shown in blue. Few more things that we can learn from this image on my slide. I wanted to mention about exons. So exons are shown as boxes, and these are over here and over here. And these, as you can see, that they could be empty boxes. So empty boxes, empty exons are non-coding, which means they're not translated, they're only transcribed. So of course, transcripts of non-coding genes, for example, in transcripts are composed of empty boxes, and then exons could be filled or colored, and these are coding. So you can see that coding transcripts are composed of filled exons. However, a coding transcript might also have empty boxes, and these would be at either 5' or 3' end, and these are called UTRs. So these are untranslated regions. So it's important to realize that not all exons are coding. And UTRs are exonic as they remain in the transcript after splicing, but they are non-coding as they're not translated. And then introns are shown as lines which connect exon, which are these boxes. And there are a couple of hints that indicate the gene orientation. Little arrows next to the gene names. So for example, this is pointing in the reverse direction, which indicates the orientation. And then above, we have these that are pointing forward. And then there's the last hint, and this is basically the shape of the introns. So if you observe these introns on the forward strand, these are shaped like hills, so they're pointing upwards. And if we focus on the reverse-stranded genes, the introns for these genes are pointing downwards, so they have this sort of valley-like shape. So we also have some annotation, some labels on the slide, which you can refer to later on. And we use two different independent annotation methods, and both of them rely on real biological experimental data. So the first is called the Ensembl automatic annotation, and this is applied to every single vertebrate in Ensembl. And the other one is called the Havana manual annotation, and this is done by a group of annotators so this is done by real people, and it's done for only human and mouse and partially for zebrafish and rat. The Ensembl automatic annotation is genome-wide gene prediction using our automated pipeline. So it's based on real biological experimental data. For this, we take known proteins, transcripts, cDNA, RNA sequencing, and we plot these onto the genome using a sequence matching technique. So people sometimes think that all we do is that we screen genomes for open reading frames, but we need to have real evidence that something is translated or transcribed to make gene annotation. And it takes about a week or two to annotate a genome using our automatic pipeline. And then we have the sources of the biological evidence that we use. So we take nucleotide data from International Nucleotide Sequencing Database collaboration, which is a collaboration between 3 institutions, including NCBI's Gene Bank, DNA's Data Bank of Japan, and European Nucleotide Archive. We might use data from closely related species. So we might be predicting genes in chimpanzees by mapping transcripts and proteins for human to the chimp genome. Then we have the manual annotation, so the Havana annotation. This is also genome-wide gene prediction, and it's done on case-by-case basis, and it's done by a person, so an annotator who moves along the genome utilizing the different types of data, including databases and papers, and it's a lot more laborious. It takes about half a day to annotate a gene, and it might take up to several years to annotate one genome. That's why it's only done for human and mouse, and partially for zebrafish and for rat for specific genomic regions of interest. And the annotators can utilize a wider selection of evidence, including the RNA-seq data of both short and long reads, and also data supporting the transcript structures like CAGE and poly-A sequencing, which can tell us something about the transcription start and end site, and also the mass spec protein and publications data. So let's talk about the advantages of manual annotation. So it is more comprehensive, especially for those genes that are difficult and tricky to annotate. So some tricky to annotate features, the non-coding features. It results in more genes in general, and also more transcripts per gene and more biotypes, especially again the difficult non-coding biotypes like pseudogenes or nonsense-mediated decay or stop-coding read-throughs. It requires less evidence sometimes, but it might be more sensitive. So if there is a small evidence that the automatic pipeline might skip the evidence without making the annotation, but the annotator can explore further and can utilize other data types and can make an informed decision based on a case-by-case basis. It's also more accurate for those difficult regions like UTRs, splice sites, assembly exceptions, and single exon genes and stop codon read-throughs. But the disadvantage is that, as I mentioned before, it's slower, it's more laborious, and it's not as throughput, so it's smaller scale. I mentioned about the transcript biotypes earlier. So one of them is the golden transcript, and these are, so we have two independent annotation methods. So as I mentioned, one of them is automatic, and then we have the Havana manual annotation. So the gene annotations for human and mouse is a product of these two independent pipelines. So we take those two lists of genes produced independently by the two methods and we merge them. And if there are gene models which are agreed upon by these two methods, so the Havana and the automatic, and by by identical, I mean that these would have the same location, the same intron exon sets, and length, such as sorry, just so for example, if the transcripts lengths are identical and they have the same location and the same intron and exon set, then these transcripts would be given the golden color. And the golden color indicates higher confidence and quality of those transcripts. Can everyone hear me properly? And please do, okay, thank you. So please also do let me know if I'm going a bit too fast or if you would like me to slow down.
There were some issues, Alina, but I think it's all resolved now.
Okay, thank you. I was just reading the chat and I could hear that some people couldn't hear me, so thanks. So now we're going to talk about the GenCode project. So it might be a little bit confusing, but the GenCode is simply Ensembl human and mouse genes. So it's GenCode for historic reasons, but the GenCode gene set is basically the merged set of automatically annotated genes and Havana manually annotated genes for human and mouse. And it's the default gene set used by many international projects that we have listed on the slide. Then another project, which is a really important one, is called the main project. And main stands for Matched Annotation for NCBI and Ensembl API. And it's another reassurance of the transcript quality. I need to start by saying that NCBI has their own gene annotation and gene set, and this is called refseq. And then the main transcripts are the transcripts which are 100% identical between these two resources, so refseq and Ensembl API. And by 100% identical, we mean that complete match from transcription start until the end, including both coding and non-coding parts of the transcript. Then we have the main select. Oops, sorry. Then we have the main select. So main select transcripts are not only identical between the two resources, but main select also provides only one well-supported transcript for every protein coding gene in human. So this is agreed to be the most biologically relevant for that gene. Also, the main select transcripts are the most representative transcripts of that locus, and they're identical between refseq and Ensembl. And by the most representative, we mean that they are on balance, they're most highly expressed, they're most highly conserved, and also span some important clinical pathogenic variants. And then very recently, the main project released additional set called the main plus clinical. There's only 43 of them genome-wide, so in very rare occasions, there might be additional main plus clinical transcripts on top of the main select. In cases where there are some clinically important variants, so these are pathogenic variants which are not captured or covered by the main select transcript. And so the main plus clinical is introduced to capture this variation. Maybe these are not most representative, but they are very important for clinically relevant variants. And then we also have the another thing when we talk about the transcript quality is another important concept, which is the Ensembl canonical transcript. So this is a single representative transcript identified in every locus. And these are basically transcripts which are on balance, are most highly conserved, most highly expressed, and these are in concordant with some other transcript scoring algorithms. And I will expand on this a little bit more later on in the demo. They are also in concordant with UniProt canonical isoforms, and they have longest coding sequence, and they cover largest number of clinically important variants, and they have to be complete. So for human coding genes, the Ensembl canonical is going to match main select pretty much. And then we also obviously have Ensembl canonical transcript for non-human species as well. There are two more concepts that I wanted to mention before we move on to the demonstration. So I wanted to talk about the Ensembl stable IDs. All of our features have assigned unique Ensembl stable IDs, and these are composed of an ENS prefix. So this stands for Ensembl. And then we have a letter that indicates the type of feature. So for example, for G stands for genes, T stands for transcripts, P for protein, E for exon, and R for regulatory elements. This is not an exhaustive list. We also have Ensembl stable IDs for gene trees, transcription factor binding motifs, and then the most important, after the ENS prefix, we have an 11-digit unique number and a versioning after a decimal point. So these are important as they are stable, so they shouldn't change between the releases. I know that numbers might not be very meaningful to researchers, and they're more likely to be working with gene names or gene symbols. However, gene names are not stable. Gene names do change, and there are many synonymous gene names. So it's always a good idea to note down the Ensembl stable ID of a gene that you're working with. And then, as I mentioned, that these are stable. So if there is a small change to the underlying feature and we have clear definition of what that small change is, we would then introduce an increment. So we would add a versioning. However, if there is a bigger change to the underlying feature, then we would retire the stable ID and issue a new one of the new feature, indicating that those two are no longer synonymous. And for some non-human species, we have their suffix added, indicating species. So we have mouse, moose for mouse, and then we have for zebrafish, we have dar. And you can find a lot more information about the Ensembl stable IDs by following this link on my slide. And then I also want to talk to you about gene ontology. So human language is ambiguous. And there are many synonyms and multiple terms for the same thing. So we might be describing exactly the same thing using different language. However, in scientific community, so for example, one person could be describing innate immune response, calling it innate immunity, and then another person might be calling it non-specific immunity. And we know that they mean exactly the same thing. However, computer searches are not going to cope very well with it. And that's why the scientific community has come up with this gene ontology terms, which are standardized, which is a standardized language, which is easily searchable, and it's controlled to talk about gene functions to make sure that we're all talking about the same thing. And all those terms have their own accession numbers. And they have a name, they have a type and definition, and they are organized in hierarchy, so this could mean that a given term, so for example, innate immune response, might have more general parent terms. So for example, like innate response, but then it might also have child terms, and there could be quite a few of them that, for example, that are shown on my slide here. And for more information on Ensembl gene annotation system, you could refer to our paper or publication on Europe PMC, which you can access through this slide. And now we're going to move on to the demonstration part of the module. Okay. So now I'm going to navigate to the Ensembl Genome browser by typing ensembl.org. And I also wanted to mention that you can find the demo on page number 22 of your course book. And I'm now going to move on to the Ensembl homepage, and I'm going to search for a specific gene by using its gene symbol. So I'm going to search for UQCRQ. And I'm going to specify my species, human. So once I'm happy with my specifications, I'm now going to click Go. So over here, now I'm on my search page, search results. And the first search hit is UQCRQ human gene, which is what we are interested in. But if you hadn't specified your species, you still have the option to restrict your search results by species on the left-hand side, and you can also restrict your search results to show genes, transcript, phenotype, or gene trees. So I'm going to select the first search hit, which is UQCRQ human gene. And now we're on the Gene tab. So yesterday we explored the Location tab, which also appears by default. And today we're going to explore the Gene tab, and later on we're also going to explore the Transcript tab. But let's focus on the Gene tab for now. So on the Gene tab, we first have our gene name, followed by its Ensembl stable ID. Then we also have some description, some gene synonym and location. And over here, under transcripts, we have this transcript table. And this transcript table, we can also choose to hide the transcript table by clicking on this blue button, or we can show the transcript table to show all of the transcripts. So we have the name of the transcripts. These are arbitrary, so you don't need to worry about these too much. The most important is the transcript table IDs. So we have the transcript table IDs in this column. Then we have the base pair length. We have the protein, the biotype of the transcript, and then we have some flags for the transcript. So over here now I want to expand on the flags. So people often ask us which transcript is the one and which transcript should they use to work with. So for example, if they want to design a CRISPR knockout, there is no straightforward answer. All of the transcripts are biologically valid, but we can provide some guidelines on choosing the most representative transcript. So if I was to choose the most representative transcript to work with, I would first look out for main select. So main select is a flag in the first transcript. And this is available for human only. And if this is unavailable, so if main select is unavailable, then I would move on to APRIS. And APRIS is another transcript scoring algorithm, and it is developed by a group in Spain. And these guys use their own judgment to score transcripts by similar focus, but also slightly different priorities. So they look at protein structure information, presence of active sites, and cross-species alignment. And they score transcripts from 1 to 5, with 1 being best. So P1 over here, which is also a flag for our first transcript, stands for principal isoform 1, and there are some additional ones as well. And then if APRIS is not available, I would then move on to GenCode Basic. And we know that we make our transcript annotation based on real biological data, and biological data might sometimes be incomplete. So if there are some fragmentary transcripts in our data, we would still make an annotation, but we would flag those transcripts as incomplete and missing at either 5' or 3' end. However, GenCode Basic filters these incomplete transcripts out. Finally, if GenCode Basic was unavailable, then we would move on to transcript support level. So these are these TSL flags, which is another scoring algorithm. And it also scores from 1 to 5, with 1 being best. We also have CCDS, and this stands for Consensus Coding DNA Sequence. This is a little bit like main, but it is older. So it's also a match between NCBI and Ensembl API, but it only looks at the CDS, so coding sequence. So only the coding portion of the transcript, and it doesn't take into account UTRs. So this is all that I wanted to say about the transcript flags. And now, if I bring your focus to the left-hand side, we have some gene-based displays. So this view shows you, lets you explore the different data that we have on this gene tab. And if I bring your focus to this sequence on the left-hand side, I'm going to click on the sequence to view the sequence for our gene of interest. I'm going to hide the transcript table for now. So now we have our sequence over here. And there are a few different things that we can do to manipulate this view. So firstly, we have the option to download the sequence. And let's click on download. So you can download the sequence in a FASTA format, also as rich text format, so RTF. And you can choose the settings of the sequence that you want to export. So you have the option to export cDNA transcript, the coding sequence, 5' UTR, 3' UTR, exon, introns, and genomic sequence. You can also choose the flagging size. So you can choose the 5' flanking sequence size and also the 3' flanking sequence size. However, we're not going to export anything right now, so I'm just going to jump out of this. And then you also have the option to blast this sequence. So you can either blast the entire sequence, or you can select a specific sequence and then just blast that selected sequence. We then have a legend for our sequence. So our exons are shown as highlighted by this peach highlight. However, our UQCRQ exons of our gene of interest are shown as highlighted peach, but also in this brown color text. Our sequence is shown as a FASTA format, which is shown in this header. And then we have its chromosome number, which is 5. And then we also have the genomic location. And we have this 1, which indicates forward strand. And then if this was -1, that would indicate that it's a reverse strand. We can also configure this page. So if I click on this blue button over here that says configure this page, this will open a pop-up box. And over here, I have some display options to choose from. So I can choose the size of my 5' flanking sequence and my 3' flanking sequence. So let's change this. Let's shorten this to from 600 to, let's say, 50. And I'm also going to change that for 3' as well. And then we can also, on this sequence, choose to show variants. So right now, this is selected as no, but I'm going to select yes, and I'm going to show my variants with links. And then you also have the option to filter variants by consequence type and filter variants by evidence status. We're going to focus, expand a lot more on the variation data and the consequence type of the variants tomorrow. And once you can also choose the line numbering. So let's also say that we want the line numbering relative to this sequence. And once we're happy with our configurations, we can simply save and close by clicking on this tick button here. This is just taking a bit of time. Okay, so now our markup is loaded, and now our sequence shows our variants, which are color-coded, and you can find the legend for these variants above the sequence over here. Okay, and now let's move back to the left-hand side menu, so the gene-based displays. And now I want to move on to the gene ontology. So this is gene function. And in the ontology section, we have these 3 categories. So we have the biological process, which tells you what the gene does. Then we have the molecular function, which tells you how it does it. And then we also have the cellular component, which tells you where the protein is located in the cell. So let's explore the biological process. So this table summarizes the different ontology terms that are associated with this gene, and we have the gene ontology accession numbers in the first column. Then we have the gene ontology terms. We have a column for evidence, annotation source, and then we also have these transcript IDs. So we have transcript IDs, but not gene IDs. And this is because the gene ontology terms are associated with the transcript or proteins. This particular table isn't very large, but sometimes they can get quite massive. So you also have the option to filter this table over here by this filter section. And you also have the option to export this table by clicking on this Excel sheet here. And the next thing that I want to explore with you is this gene expression option that we have here. So I'm going to click on gene expression on the left-hand side. And this is going to load external data from the Expression Atlas. And we have this really nice interactive widget. So you can change between sexes. You can also choose different brain locations by clicking on this brain icon here. And it is all interactive. So you can hover over it and select the different tissue that you're interested in. And we have this heat map over here on the right-hand side, which summarizes gene expression across different tissues, and then we have the different projects. And then if I scroll to the bottom, we also have a legend for our heat map. So high expression is a darker blue color and low expression is this light blue. And the last thing that I want to show you on the Gene tab is this external references on the left-hand side. So I'm going to go to external references. So on this page links you out to other databases and resources, which also have some information on our gene of interest. So this page will link you out to, for example, Expression Atlas, OMIM, NCBI gene, and Reactome gene. And then it also has some IDs which correspond to our gene of interest and its transcripts in other databases. So it's a really nice and great place to branch out from. And now we've covered some of the data that we have in the Gene tab, and now we're going to explore the Transcript tab. So to go to the Transcript tab, I'm going to click on Show Transcript Table. And from this, I'm going to choose the first transcript. And I'm going to click on this transcript ID. And this is now going to open the Transcript tab. So the Transcript tab also gives us some information, and this information is the same information that was available on the Gene tab. And then we also have the transcript table. And then in the summary section, we have the graphical representation of the transcript and some information about this transcript. So we have the statistics, so we have the number of exons in this transcript, we have the UniProt identifiers, the version of the transcript, the type of the transcript, so it's a protein coding transcript, and some more information. And then on the left-hand side, we have some transcript-based displays. So I'm going to focus on sequence again, and over here, the sequence is also, we have 3 different categories. So we have exons, cDNA, and protein sequence. I'm going to click on exons. So here we have a table summarizing all of the exons in this transcript, and you can find the exon, we can find the exon stable IDs. We also have the start site, the end site, start phase, end phase, the length of the exons, and then we also have the sequence of the exons. We have a legend above this table. So the translated sequence is in a blue text. The flanking sequence is shown in this green text. The intronic sequence is shown in gray. UTRs are shown in orange. And similar to other sequences displays that I showed you on the Gene tab, we can also configure this page. So let's configure this page, and I'm going to show you the different options. So over here, we have the option to change the flanking sequence at either end of the transcript. We also have the option to show full intronic sequence as by default. This is truncated in our actual display. And then you have the options to show exon only. You can show variants and a lot of other different options as well. So I'm not going to change anything over here. So let's just step out of it. And then we also have the option to download the sequence. And you can download the sequence in the FASTA format or RTF, so rich text format as well. And then we also have some additional settings. So you can include in your sequence the 5' UTR, 3' UTR, exons, introns, and the genomic sequence. And you can also choose the size of your flanking sequence. Okay. Now I'm going to go on to the cDNA sequence. So this is the sequence of a mature splice transcript. So our cDNA sequence, this view is composed of 3 rows, as you can see that we have these 3 rows here. So the first row is our actual cDNA. So this is the splice transcript with UTRs, which are indicated by this yellow highlight here. And then in the second row, we have the coding sequence. So this is the CDS, and it starts over here. So it says indicated by 1, and it goes on in this second section here. And this starts with, as you can see, it starts with A, T, G, and it finishes with TGA. And then the third row is actually a protein sequence, starting with methionine, and then it finishes with a stop codon. Exons over here are shown as blue and black lettering. So these are alternative black and blue. So you can see that the first exon finishes about here, and then the black lettering shows the start of the second exon. And this is it that I wanted to show about the cDNA sequence. And now we're going to explore the external references. So in the external references section, we have 2 categories. So we have the general identifiers, and then we also have the oligoprobes. The general identifiers will show you similar information that we also saw in the external references on our gene tab. So we're going to click on oligoprobes. And this page shows you microarray oligoprobes mapping to this transcript, and these can be used to measure the expression of this particular transcript. And the last thing I want to show you, I want to bring your focus to, is the protein information. So proteins are not features in Ensembl. So they don't have a dedicated tab because they do not map to the genome directly. They map to the genome via transcript objects. So you can find a bit of protein information on the transcript tab, and this includes protein summary, domains and features, variants, and 3D protein model. So let's click on protein summary. So over here, this page shows you a graphical representation of this translation. And then over here, we have the N terminus, and then we have the C terminus. And this translation is colored to show you exon boundaries. So you can see that this pink color is the first exon, and then the second exon has this purple color indicating that they're different. And then we also have some protein domains predicted by different methods. So these are all in silico predictions, and these are not experimentally validated. These different methods do not always agree, but if a given protein domain or motif are independently predicted by various methods, it's most likely to be real. You can also find all of this information summarized in a table format if you're not a fan of graphical representations, and we can find that in domains and features. So I'm going to click on domains and features on the left-hand side. So now over here, we have the same information in a table format. So we have this column over here for domain source. We have the start and the end positions and the description. And then we also have some accession numbers as well. And the last thing that I want to show you on the transcript tab is the 3D protein model. So I've clicked on the 3D protein model on the left-hand side on the transcript-based displays. And over here, we have our 3D protein models, and these are from PDBE. And we have this nice interactive widget, so you can click on it, and you can also rotate it. So you can manipulate this view. And then on the top here, we have some information about the PDB mapping. We also have some information about the exons, so you can choose to see the exons or not. And then you also have some protein information, which you can choose to view. Let's just bring it back to original. So over here, so you can play around with it and view different protein informations. And then we also have some information on variants, so SIFT and polyphen, which are pathogenicity prediction scores that you can choose to view. So it's quite colorful and I quite like it, it's nice. And this is it that I wanted to show you about the gene tab and the transcript tab. It is a lot of information. So I think now you have time to explore some of this information by yourself by doing the exercises. And the exercises are on page number 31 of your course book. And if you have any questions, please don't hesitate to ask us either on the chat or on the living document. And we'll give about, I guess, about 20 to 25 minutes, depending on how you're finding the exercises. And then if you have any questions, I'll be happy to show you on the screen. So there's a question about part B of our exercise 1. So let me just jump out of my presentation and go to the course book. So part B has asked for click on phenotypes at the left-hand side of the page. Are there any diseases associated with this gene according to Mendelian inheritance in man? And we're exploring the human MYH9 gene. So let me just copy this. Never mind. I'm going to go back to the Ensembl homepage and I'm going to search for the MYH9 gene in human. And I'm going to click go. And now I'm going to select MYH9 human gene. So part A has asked us find the human MYH9 gene and go to the gene tab, which we've just done. On which chromosome and which strand of the genome is this gene located? So we can find information about the location under the gene name and its stable ID. So over here we have this location line, and this gene is located on chromosome 22, reverse strand. And then how many transcripts, splice variants, are there and how many are protein coding? So we can find information about the transcripts by showing the transcript table. So we can count all of these transcripts. And we also have this information about this gene line, and this gene has 11 transcripts. And the protein coding information is given in the biotype column. So we have 3 protein coding transcripts. And then the exercise asks us about what is the longest transcript and how long is the protein it encodes. So we'll go back to our transcript table. So we can find information about the longest transcript in the base pairs column. So the longest transcript is MYH9201, and then we have its transcript stable ID, its base pair length, and then we also have the protein length in this protein column. And then it asks us about which transcript would we take forward for the study. So for this, we're going to focus on the flags associated with the transcripts. And I would choose the first transcript because it has the main select. It is also the Ensembl canonical transcript. It also has the GenCode basic flag, and it's P1 and also scores 1 for both appraise and TSL. And now part B, we have to click on phenotypes and then find for diseases associated with this gene according to MIM. So let's do that. So I'm going to select phenotypes on the left-hand side. And now over here we have a table that lists all of the phenotypes, diseases, and traits that are associated with our gene. And to find the OMIM source, we can just filter this table and type MIM over here. And now we have all of the phenotypes. And now we have all of the phenotypes that are sourced from the MIM database. There are a few questions about the untranslated, so the UTR sequence in the exons for the first exercise. So let me go back to the MYH9 gene, which is the human MYH9. And part D of the exercise has asked us to click on the first transcript. So I'm going to click on its transcript ID, and this will take me to the transcript tab. So now on the transcript tab, in the summary section, we have some statistics about the exons, the coding exons, and some more information about the type of this transcript and the annotation methods used. And to find the exon sequence, we're going to focus on the left-hand side and click on exons. I'm just waiting for it to load. Okay, so our exon sequence has now loaded, and you'll see that the flanking sequence over here is given in green. And then we have the ensemble stable ID for all of the exons. So within these exons, you'll see that in exon 1, it is a complete UTR sequence. So this is shown by orange. We also have the legend over here above the table. And then exon 2, some of the sequence in exon 2 is a UTR, and then the rest of it is translated sequence, which is shown as blue text. And then if you scroll down, the rest of the exons have the blue text, so they are translated. And then a major part of exon 41 is untranslated. So this is shown here as it's shown in orange text, so it's UTR sequence. I hope this answers your question and clarifies some issues. Anisha, so I think what's happened is that when you view the exon sequence, you also have the variants marked on your sequence. So it is the same page, you're on the right place, but you can go to configure this page, so this blue button here. And from here, in this show variants line, you can select no. So we have this option of no, yes, and in exons only. But if you don't want to show any variants, so it will just make the sequence look a bit cleaner. So just choose no, and then save and close. So it's show variants, no, and then save and close. And then this will load a similar view to what you're seeing on the screen right now. It's now time for us to end today's module. And today we've spoken about genes and transcripts in Ensembl. I spoke to you about the two annotation methods and the pros and cons. So that's the automatic pipeline and also the manual Havana pipeline. And then I also showed you how you can navigate to the gene tab and the transcript tab and the different types of data that you can find there and how you can manipulate the sequence views and how to export them. And also, if you want, you can also blast the entire sequence or a selected sequence. Tomorrow, we're going to focus on variation data in Ensembl, and I will speak to you about where we import our variance